The disease, X-linked adrenoleukodystrophy, it's a rare orphan disease, and it's a paroxysmal disorder. So I'm giving a brief introduction into these paroxysmal disorders. Basically, they can be classified into two major groups: the paroxysmal biogenesis disorders and the single enzyme deficiencies. The paroxysmal biogenesis disorders, and they results from the peroxin proteins, which are required for the construction or the biogenesis of the peroxisomes. PEX5 being the most common of all those. And so whenever there's a mutation or a deletion in these proteins, you basically have non-functional peroxisomes, also called as the ghost peroxisomes. And some of the diseases characterized by these are Zellweger and RCPD. And there are other single enzyme deficiencies in peroxisomes, the X-linked adrenoleukodystrophy being the most common and the most frequent peroxisomal disorder with a single enzyme deficiency. So the gene in this case is called the ATP cassette binding cassette family D1 or ABCD1 gene. And the common name also is known as ALDP because that's the protein it codes for, the adrenoleukodystrophy protein or the ALDP. ALDP is an integral membrane transporter protein. It present in the, as you see in the second figure, in the membrane of the peroxisome. And it is responsible for import of very long chain fatty acids, saturated very long chain fatty acids into the peroxisome for degradation. These fatty acids saturate very long chain which have carbon chain greater than 22. This function is exclusive to the peroxisomes. So these very long chain fatty acids cannot be beta oxidized or degraded in any other organelle in the cell. So as a result of this mutation or deletion, you basically have non-functional, or in 75% of cases, you have absence of ALDP in the peroxisomal membrane. So this results in accumulation of very long chain fatty acids in the body fluids and tissues, including the CNS. So although it is a single enzyme deficiency, and you see what will look like is just the accumulation of very long chain fatty acids, but the disease gets much more complicated from here. So basically, you have two stages in this disease. So the initial is the metabolic disease, which starts in utero in the absence of ALDP. And you can say this portion is common to all the patients of XALD. All of them will accumulate very long chain fatty acids, which you see. The top portion is the normal patient, a normal human being, and the XALD patient shows all the very high fatty acid, saturated fatty acid peaks. But in about 30%, 35% of the patients, this disease spontaneously progresses into a CNS inflammatory demyelinating disease. So you start seeing demyelination in the brain, inflammation, and within the initiation, within 2 to 5 years of the initiation of this brain inflammation and demyelination, the patient dies. This inflammatory portion of the disease is most common in children ages 5 to 12, and so most of them will die before the age of 12 once the disease initiates in the CNS. Whereas the AMN patients, they live up to 40, 50 years of age, and the only pathology in them is seen in the spinal cord, distal axonopathy. So they have mild motor function, motor dysfunction in later stages of later ages. However, what causes this transition remains unknown till today, and it is further complicated by the fact that there are about 15,591 mutations known as of today in ABCD1 gene, and none of them correlate with any particular phenotype. So much so that within the same family or even between the twins, the same mutation, one may have AMN, the other may have the ALD phenotype. AMN, I think I didn't mention, is called adrenomyeloneuropathy. And further complication arises from the fact that the mouse model deleted for ABCD1 only accumulates the very long chain fatty acid, never develops the CNS demyelination or inflammation. So I have been focusing until recently on the proteomics aspect of the disease, and during those studies, one of the things which we found was this gene AMPK, AMP activated protein kinase, which is a metabolic gene, but it's also known to play a role in anti-inflammatory role. So it has been shown that under inflammation, its expression or activity goes down. So what we found was, surprisingly, in fibroblasts and lymphocytes derived from XCLD patients, we saw that this AMPK was significantly down. And the metabolic function, which was measured with the CHORS, as well, the overall metabolic profile was down in these ALD patients, but not in the AMN patients. So this is where from we started to think about the metabolomics and look at metabolites, whether they can be differentially altered. Specific focuses between AMN and ALD phenotypes. So the overall experimental design for this study is what you see on the left side. So this grant, which is supported from the Michigan Metabolomics Core Facility, is right now we are focusing only on the global metabolite differences between AMN and ALD. But what I have summarized here is our long-term goal of looking at those metabolites and validating them and then forming a hypothesis once we have all the data. So what I'm presenting today is basically fibroblast data. This is an ongoing grant, and right now the AIM2 is being worked on. So we took the fibroblast from control healthy patients, AMN and ALD patients. These are the skin fibroblasts, and provided them to the core facility. So based on the data, we could clearly see that the metabolites between control AMN and ALD were clearly separated in the three samples. So when the analysis were done by the bioinformaticians, we came up with, so this is when we kept the significance at 0.05, we got these top 7 to 10 metabolites, and among the top was glutathione metabolism and the other pathways. However, if we made the conditions a little less stringent, we could include about 19 to 20 metabolites, but this is what we've right now focused on. So when you construct the heat map and you see a significant, so now in this, I would like to point out that this glutathione and glycerophospholipid metabolism, it kind of confirms whatever hypothesis is there currently in the XCLD field, because glutathione metabolism or oxidative stress is right now considered the first hit in the progress of the XCLD disease. So it was kind of reassuring that we are kind of able to find out those metabolites in our study in going forward. So when a network was constructed from this data, so the top network that we found showed basically changes in cell cycle metabolites or pathways involved in cell cycle or DNA replication and repair and the molecular transport. So this data is being, this is only a part of the data, and the data is still being further analyzed from this study. So overall, what we concluded from the partial grant which we have completed is that there is a significantly better discrimination between the three groups. So what is important is that we can identify some compounds or some metabolites which are differing between AMN and ALD. This is kind of a significant step forward, because right now the only diagnostic test for XCLD is to look for very long chain fatty acids, which are same between AMN and ALD. So when a patient is born, even if there is a family history, you look at very long chain fatty acids, you know he is an XCLD patient. But whether he will go on to be an AMN or die within 5 to 10 years by ALD, you can never predict. So So the fact that there were some biological markers which are already known to be changing in like the glutathione metabolism are appearing in our metabolite study, that makes sense. And the comparison between AM and ALD again is novel, and we hope to find further good data or good leads in this study. So the AM second, which is currently ongoing, we are looking at the postmortem brain white matter. So in this study, what we are doing is we are taking only the XCLD brain. So within the XCLD brain, the demyelination and inflammation is very focal. So you see focal areas of demyelination and inflammation, and you see areas of white matter which are completely normal. So they have white matter, they have very long chain fatty acid accumulation, but they are completely normal. And then you have the areas called shadow plaque around the plaque areas, which have inflammation, demyelination somewhere in between the normal looking and the plaque area. So this is, we have already published about 3 years back when we looked at different parameters of CPLA, basically 5-LOX pathway in that study. So we are taking that brain and we are providing the tissue, we are providing the samples from normal looking area, plaque shadow, and the plaque within the same brain. And we are going to look at how the metabolites are changing, because the ABCD1 mutation is common, the very long chain fatty acid between these areas are common. So what are the metabolites that are changing between these 3 areas that lead to the progression of the disease? So that is the study which is currently going on. And this group, in my group, Brittany is the research fellow working on this, and our collaborators, Shailendra Giri and Ramandeep Ratan, they provide critical comments on the data, and Laila did all the bioinformatic analysis for this study. And the study is supported by Michigan pilot and feasibility grant. Thank you.
Any questions? So did you see any glutathione conjugates in your metabolite studies? Yeah, the microphone. Yeah, did you see any, and was glutathione being conjugated with any of these metabolites that?
No, we have not looked into that.
Okay, and did you, and by any chance, were there any increases in dicarboxylic acid since peroxide?
No, there was not. Actually, the glutathione metabolic, glutathione or glycerophospholipids were the major which were coming up in these analysis.
Any other questions? Thank you.